Well, hello, class. Back again for our third lesson on evolution. In our last lesson, just to recap quickly, we talked about some methods that were used to study evolution, to study the age of rocks and the age of fossils. And these included studying the fossil record using the principle of stratigraphy. That is, the more deeply buried the fossils are, the older that they are, and also supplementing that with radioisotope decay measurements using such things as carbon-14 or uranium-238 and other radioisotopes. We also just briefly mentioned some more familiar techniques like dendrochronology, which is measuring the number of rings on a tree. Of course, that you don't go that far back when measuring live plants. Amino acid racemization, DNA mutations. And in this lesson, we're going to talk more about genetic methods to study evolution. And we'll move then towards vestigial organs and how that gives us evidence for adaptation of species. Now, let's begin then by looking at some genetic methods and how they are used to help us understand evolution. Cytochrome C, we'll focus on a protein. This is a protein found in all, or essentially all, eukaryotic organisms. It's called cytochrome C. It's not so important what it does. You see a drawing of cytochrome C in the bottom right. This is one of the ribbon drawings. Of course, cytochrome C is a typical protein. It has about 100, 105 actually on average amino acids that are strung together in a long polymer, and it folds up into a three-dimensional structure. Its function is to carry electrons from one species to another. And it's found in almost all eukaryotic cells. So it's an important protein. And it's a fairly small protein and one that's easy to study. What scientists have done is determine the amino acid sequence of the cytochrome C, of this protein, from a large number of different species. Of these 105 or so amino acids, it's kind of interesting that 38 of these are invariant, meaning that they are the same amino acid in the same position, like position 13 or position 82. It's the same amino acid in all species that have been studied. But the other amino acids, the 105 minus 38, can vary from one amino acid to another. And in fact, there are 8 positions that are what are called hypervariable. That is, most any amino acid can be in those 8 hypervariable positions. So amino acid sequences can be determined, and if you do this for a large number of species that you're interested in investigating, now this slide shows a lot of letters and a lot of colors, and it looks like it might be a little overwhelming. But if you look over to the left, they studied the amino acid sequence of this protein, cytochrome C, from a number of mammals: humans, chimpanzees, monkey, horse, donkey, cows, rabbits, kangaroos, and then other vertebrates, pigeons, ducks, turtles, rattlesnakes, bullfrogs, tuna fish, then some insects, and then some fungi, and then some plants, including wheat and buckwheat, pumpkins, cotton. Each row represents the amino acid sequence of this protein, cytochrome C, from the species, and this is the one-letter abbreviation that we've talked about. Now, it's hard to see because there's so much information here, but if you could look at some of these columns, such as 30, it has this a P, a symbol P, which happens to be the amino acid proline. It must be one of these invariant positions because it's P in every case. But then if you look at some other columns, some other positions, you can see that there would be some different letters, that meaning different amino acids at different places. So by looking at the amino acid sequence of a number of different species, one can make comparisons. And this is typically done in a manner shown in the following slide, which again looks kind of complicated, so let me try to explain it to you. Essentially, this is sort of like a two-dimensional representation of the number of differences in the amino acid sequence when comparing two different species. The arrow will point to man and chimpanzee, and if you then go up this column, you would see that it lists the number of variations between the amino acid sequence for this protein for man and all of these other species. And of course, if you go to the top where you compare man with man, it would be 0 because you're comparing something with itself. But if you compare man with the rhesus monkey, there's 1 variation. With the horse, there's 12. With a donkey, there's 11. So you get the point. That is, if the number of differences is large, it suggests that these species are much different from each other, which in some cases you would say, well, that's pretty obvious. But it will also give us information about when species diverge from each other in a phylogenetic sense, enabling us to construct genetic-based phylogenetic trees. Much like, and this is shown in this slide, much like the phylogenetic trees that we've seen before, which were based on physical characteristics or phenotypes. But this is a phylogenetic tree that is constructed based on molecular information, that is, the sequence of amino acids or the sequence of DNA for the gene for cytochrome C. And so in this phylogenetic tree, you can see that over to the right, things like Neurospora, which is bread mold, or Candida fungus, that these diverged early in an evolutionary sense from humans. Humans are at the upper left-hand corner of this slide. And you can see the various relationships between these different species. Now, analysis shows that the cytochrome C gene for humans is actually identical to that for the chimpanzee and very similar to that for the monkey. It's 86% homologous with the rattlesnake and 50% homologous with yeast. So the more homologous, the more similar the species are, and the less homologous the amino acid sequence for cytochrome C, the more divergent these species are. Now, the next topic we want to talk about is vestigial organs and genes, and how organs and genes have adapted. Now, vestigiality is when organs and genes have apparently lost most or all of their ancestral function in a given species. Now, a typical example, the wings of an ostrich. An ostrich can no longer fly, but it has wings. The wings of a penguin. A penguin can no longer fly. The leg and hip bones of a whale, vestigial organs, because the whale doesn't have legs. The human appendix, an organ which no longer seems to have a function. Also, the human coccyx or tailbone, which is a vestige of a tail, which of course we do not have. An interesting example is the enzyme that makes cytochrome C. Its name is guanylactone oxidase. It's a complicated name, but it's an enzyme that will make cytochrome C. It turns out that humans and a number of other primates have this gene in our chromosomes, but it's inactive. And that's a reason why we have to have vitamin C as a vitamin. We have to have it in our diet because the gene is no longer expressed even though it's in our chromosomes. So we've lost the ability to turn on or to activate this gene. Now, a reason that vestigial organs are interesting is because they also show how adaptation takes place. An organ or a gene that once had a function may be adapted to a different function. The wings of a penguin, for example, no longer enable the penguin to fly, but they're used for swimming. The adaptation of arms and feet to be wings in birds and bats. And to the right shows homologous organs and how they have been adapted in different species for different functions. An interesting or a curious example that is discussed in the field of evolutionary biology is the laryngeal nerve in the adaptation of the giraffe. Now, this seems like an odd topic, but it's kind of an interesting one. The laryngeal nerve controls the voice box, which we need for speech and for swallowing. And the laryngeal nerve connects the brain to the larynx or to the voice box. In fish, the laryngeal nerve controls gills. But the brain of a fish is fairly close to the gill. They're located next to each other. But in animals, there's a slight difference in the location of our brain and our voice box. But in some animals, particularly mammals, that evolved longer necks, and of course they evolved longer necks so they could eat the fruit in the leaves and the trees. As they evolved longer necks, something interesting happened. It turns out that there are two nerves for controlling the larynx. One is it's right or left, or superior and inferior laryngeal nerves. One of the nerves goes directly from the brain to the larynx, but the other does something funny. It loops down around the heart, around an aorta coming from the heart, and then back up to the larynx. Well, again, in most mammals, the distance between the brain, the larynx, and our heart are not very far. But as a mammal evolved a longer and longer neck, what is curious is that this one nerve goes all the way down from the brain, all the way down, looping around the heart, the aorta coming out of the heart, and then all the way back up to the larynx, several meters, just to innervate or control the larynx. So again, this is just an interesting example of adaptation and the consequences of adaptation, the giraffe evolving a longer neck, and how the path of the nerve did some funny things in order to adapt to this adaptation. Okay, now the next time we're going to talk a little bit more about the genetic basis for adaptation and leading to the concept of natural selection. So we'll see you in a while. Let's cue up the piano.